Cool. Thank you everyone for being back very punctually. And now it's my great pleasure to introduce Sid, who I guess also doesn't need to be an introduction, as the final speaker of the conference. Thank you, Sid.
Okay. So talk about cell fate dynamics. Most of this work was done, some of the early work on reprogramming was done with Nika and Sophie. Some of the theory work was done with Madhav and Simon, and Spencer is a postdoc in the group right now. So I guess I'm just going to set up the problem, which we have done many times now. You know, stem cells going to progenitor cells of different kinds, making different sort of differentiated cells, and we could put a landscape below it or not. Depends how you feel about it. But then there's also sort of this idea of reprogramming, right? Things can go back, and clearly, at least, there's no sense of a landscape for stuff going backwards, as much as we know, right? That remains a question in some ways, right? We kind of hit it in the sense, at least going forward, we kind of handhold the cells with lots of signals and bring them down in a very sort of canonical way. Going back, we kind of hit them with 4, 5, 10, what's your favorite number of Yamanaka factors, and then, you know, some tiny fraction of them turn into reprogrammed cells, right? So again, the question is, you know, in many contexts, we are starting to realize that it's not one stem cell. There's already sort of this plasticity, gene expression level plasticity of the stem cells. And at least in context of blood regeneration, people have seen that some stem cells make more myeloid lineages, some make more granulocyte lineages. So there's already a sort of a pool here, which is not homogeneous, which may play a role in terms of how we think about sort of this new cells coming out from the pre-existing ones. And so on and so forth. So there's also a diverse pool here. When you want to think about reprogramming, does that play a role? Those are the two questions I'll start thinking about. So just as an outline, I'll start by setting up the problem with reprogramming, because I think I like the setup in that way. This is older work though, but there was something interesting we found which led us to think about more in terms of how we can think about these cell fate dynamics. In the second quick bit, I'll talk about, this was work with Madhav and Friedman, Simon, in terms of thinking about, can we look for these so-called bifurcations, as we have been calling them, in context of actual single cell data, now that we have a lot of it. And finally, can we think of a little bit of a regulatory logic in the sense of, you know, as Anita was also mentioning, and we have been discussing throughout this meeting, that these are self-made signals, right? So in a dish, you can do sort of signals from the outside, but in a developing system, there's sort of intrinsic signals. So can we look for that kind of thing when you have this kind of single cell data? Okay, so cellular reprogramming. So I think this started with a sort of a simple idea where people knew a lot about, when we started thinking about it, that these things can reprogram. And there was this clear sense that, of course, a skin cell, in this case fibroblasts, are very different from pluripotent cells. Many, many genes change. And people had sort of some sense of that there might be many paths these cells are taking back in this gene expression space. They had kind of analyzed something what they call pre-IPS states. This is sort of like a state, I think Mohit was mentioning, in the sense you give the signals, the state can persist as long as the signals are around. But if it has kind of passed this, you can take the signals out and it'll move on to this IPS state. So there's sort of some kind of intermediate state which has to be pushed up to before it can go down on the other side, if you want to make a hill type analogy. But I think what was interesting was that now if I look at, just want to sketch out this experiment. So you can take single cells and reprogram them. Or you can take a population of cells and reprogram them, right? Again, the question is, you know, does the signaling or interactions matter or not? And you can do sort of a very simple experiment to start with, which is not many cells, but you do find, so these two axes are basically number of cells which are sort of IPS versus pre-IPS, right? So each dot, green dot, comes from a single cell. So you take a single cell, you reprogram, stuff is growing, then you ask what fraction of your cells are pre-IPS versus IPS. And you see a huge sort of diversity in the sense of single cell behavior. Yeah. Yes. So basically nothing happened, right? Yeah. So yeah, they don't add up to 100, I guess. Yeah. Yeah.
And just to be clear, is it important that it was a single, like one cell being reprogrammed, or a clone of cells being reprogrammed?
In this case, it was a single cell. And then you just basically look at the clone that has grown and sort of separate it out in these things. And it's sort of, marker type analysis. But if you take a population, basically you see a very tiny fraction of pluripotent stem cells. Right? Most of them are actually stuck somewhere here. So this was sort of the motivation, it's like a percent type yield. And those are the kind of yields people see in these experiments in general. So this was sort of the motivating idea of, single cell versus population, and we wanted to think a little bit more about it. And Nika came up with an interesting experiment which was done in another group. Okay, this is basically just saying is reprogramming sort of more development-like or these group behaviors. What she basically did was barcoded these cells, right? You can take a large population, but barcode them, right? And because they're barcoded, you can track their clonal lineage, right? You can't track individual cells, but you can track the clonal lineage, what came from what, right? And the idea was to basically now, because you have clonal lineage, maybe you can separate out this sort of heterogeneity if it's there and what might be there. So we didn't get to that richness, but, and these experiments are long. So when you start, you can sort of, you let them divide for a few times, so you get sort of roughly an exponential distribution. So this is, should have plotted better. So this is just fraction of clone sizes of different size, right? It's a relative clone size. So you see a bit of a breadth, but not that much. Some are divided, some have divided a little more, others less, but it looks like it sort of is an exponential distribution as you would expect from a random birth-death process. And now you can go forward in time and kind of look at these distributions again, what happens to them in time. And the simple idea of things being homogeneous clearly don't work out. You really see they're much bigger tails in the sense that they are cells, they're clones which are much bigger, and these are log scale. So these clones can be sort of hundreds of times bigger than your average, if you will, right? Yeah. So I'll come to it, but it comes out to be a few percent, right? Because it goes in the log, right? So you can estimate those numbers, right? So this is exactly what we did, like how do you think about these populations? Are there multiple types? But again, the data is not that rich. So what we did was basically made just a 2-population model, right? Saying that you have some things which grow much faster and reprogram much faster. And I didn't put a plot here, but I think the average was around 5% or something. So 5% was, so now you basically need 2 parameters to fit it, right? One is the fraction of these cells which grow faster and reprogram faster, and what is their growth rate difference, relative growth rate difference, right? Those are the 2 numbers you can fit to get these sort of black lines, which tell you that at least you see these cells which can reprogram much faster than, yeah. Yeah. Right.
And then there's this other population, and there's a transition rate.
Got it. Yeah. Correct. No, but there's more here. What this is saying is that what comes out from these skin-like cells, those 2 stuff that is coming out may also have differences in their growth rates. Yeah, right. And so skin-like cells are reprogramming, and the question was, in these reprogrammed cells, are there differences, right? And that was important to understand this sort of, yeah. No, it's good.
But in the previous problem, you showed the percentage.
Yeah.
Added to 100%. So there are some cells that.
There's some cells left as skin cells. Those are not counted here, so we sorted them out and just looked at the other ones. Yeah, and the skin cells actually don't grow that much, so it wasn't a bad, yeah. So they are there, but they don't grow that much. Yeah.
So when you say competition in vitro, that's just coming from growth rate differences.
Completely, yeah. But this is also reprogramming, right? So they're also reprogramming. So what we don't know is whether it's the rate of reprogramming or the growth rate differences. That's kind of pulled in, right? Because I can't tease that effect up, right? Yeah. Okay, so now you've fit the model, which is great. So we wanted to ask, does it make some prediction whether this holds true? So this was another experiment, I think, again, Nika came up with it. She said, why don't we do a splitting experiment, right? You can take the cells and just split the 2 pools and run them in parallel. And you can ask whether if there is sort of something, better, you can inherit it or not, right? So these are kind of correlations. You see that, of course, this is at T0 where you've just split it. So up to noise, basically you have bigger clones in both of them. Same clones which is big in both of them, right? And here you start seeing sort of a spread, right? And this is just overall correlation between these two flasks, right? And here what I'm showing is basically the green was the measurement, the correlation measurements at different time points, and the squares was what our model would fit, right? So these are not new parameters. So based on the previous model, which tells you what the two growth rates differences might be and what fraction of them might be, would predict some correlation between these things, right?
This is some correlation in gene expression space?
No, this is clones, sizes of clones, right? So right now there's no gene expression, right? All I can track is lineage of clones, right? So this is just saying that, if a clone is big in one, the clone is going to be big in the other, and by chance you won't get it, but if you have this structure of some things growing faster, that's going to increase correlation as you would imagine, right? And so that's what these squares would give you. So at least it says that, and then you can also test other aspects of the model, which is basically how much diversity do you preserve over time, right? So those things are also kind of explained by these two population models. So at least here we didn't have to invoke new parameters. Basically, once you have those two, you can kind of explain other properties of this population dynamics that is happening. So what this gave us was sort of this picture, which is not fully cleared up yet. The picture is that, you have some skin cells. Now I'm trying to do some gene expression space, right? You have some skin cells, you have some pluripotent cells, and it looks like there's some sort of cells which take paths which take them much faster to this end state, right? And the others are kind of floundering around, right? So at least we have two hypotheses which we want to test, and we're trying to do new experiments. Is this sort of happening because of new signals happening in the population? Or that actually genuinely these paths exist and they're development-like. And if you find them early on, you kind of just go quickly, and the others are just lost because you are kind of hitting it with a hammer in this high dimensional space, right?
So these floundering ones that you
Yeah.
mentioned, I mean, where do they eventually?
I mean, in the experiments, so there are some single cell experiments I'll come to towards really the end that some people have done. Most of them die in a dish. So nothing happens to them.
They don't come back.
They don't come back usually, right? Yeah.
A similar question.
Yeah.
I missed, I guess, that part of the design. So what has the rapid growth got to do with stem cell-ness?
Right, so skin cells don't grow. Once they pluripotent, once they switch to that stage, they start growing, basically.
As soon as they start growing, you assume that they can, or are they on their way to being?
On their way, so basically, whatever experiment suggests is that they can, once they pass this pre-IPS-like state, they can actually grow quite quickly. And pre-IPS also grows a little bit, yeah. So you're right, so there is, You could. And it turns out I don't show this. So what we did was basically precisely this idea. You can ask how much the heterogeneity at each stage predict the final stage. So day 0 doesn't do much, but by day 8, you actually start predicting a lot. So there's some early events which kind of decide pretty much which ones are going to make it to the end. So that's the reason we kind of, so, but is it still signaling, which is allowing you to reach these sort of early ones, or sort of cell intrinsic sort of randomness that I don't know. Any other questions? So I guess the questions I just said sort of now led us to thinking about, can we think of these as dynamical systems which we can learn properties of using single cell data, right? So this brings me to the second part, which was sort of not going to be, I'm going to come back to reprogramming in the end. I just wanted to show an example of looking for these bifurcations and how probably one can do that in single cell data. So this was in context of blood. You make lots of blood cells of different types. And we wanted to ask, is there dynamics in gene space that you can look at? So this was this amazing data set came from Alan Klein's group in 2020, where they basically had two data sets, one in mouse and one in a dish. I think their goal was to basically look at how cells, how much can you reproduce in a dish. They were also doing sort of lineage tracking in it, but in this experiment, they couldn't track that many lineages, and there was other technical reasons for it. But the question we wanted to ask is, well, we make these colored maps of different cell types defined in many ways. So clearly there are some transitions happening from an undifferentiated cell to these cell types, right? And our null model has been these bifurcations which take us through them, right? That has been the model that we have in our head. So we're like, okay, can we kind of look for them if it's even possible in this kind of data? So we started with one trajectory, there are many cell types here coming out of this sort of blue mass. We said, we'll stick to neutrophils, and neutrophils were the one which you saw chasing this bug in one of the videos. And you can kind of stack them in time and say, which was later, which was earlier, right? And this, yeah.
This is not really a time series.
This is not a time series. Very good, right? So here you take a snapshot. I mean, in this case, what happened is because they also were doing in a dish, they could get some sense of time series. So one can inbuild that in, but in general not, because even that is too coarse to really say that we can use that to put cells on them, right? So even in the experiments, when you see that, it's sort of hard because they're so sparsely resolved, right? Reprogramming, you can do that because things actually kind of move together, but come back to that. So I guess the question was, do you see this? And of course, genes are changing a lot. Along this path, this is just sort of looking at different, 400 odd genes, which are changing a lot during this process. Very good. Okay, so here the idea was, I think there are many ways people have done this, so this is not we have thought about, and I'll come in the next part where we try to do a little bit better. Here, the idea is basically you're looking at a cell, growing a circle around it, finding a cell which is closest, given it the next timestamp, and so on and so forth. You can do a little bit better because now you can also assign little vectors to each one using spliced and unspliced. So that has kind of made this a little bit better, but that's what basically this tiling is. 0 to 11. No, that one is basically just saying spliced, unspliced, right? So an RNA comes out, it gets spliced. So if you see a gene which is spliced, that must have come later than, yeah. So it gives you a direction for where it's going. So you can use these vectors and where it is to kind of stitch them together. That's what most of the algorithms do. I think some people actually probably know more about it in that way. Yeah?
Just a main question, but in the previous slide you have these types, those are all labeled by hand, right?
Meaning you again look at this gene expression to look at defining these, right? So in this case, it's nice because they actually have many, many markers for these things. This is not looking for new cell types as a cluster or something, right? Yeah, so these are very well-defined cell types. So that's why it's a good system in that sense, right? So yeah, I'm sure there's some hidden new cell types which people don't know about, and in some places they look at. Here they are kind of well-defined. So you can clearly see that the blue mass is sort of very diffused and people don't know about it, but these sort of, these tips are well-defined in that sense, yeah. So you can see lots of genes changing. So if you wanted to really think of some kind of, sectors of, genes that are changing together or something, which would be important to think about these bifurcations, how do you do that? So basic linear theory again helps us, because, this is sort of, so imagine this, that you take cells and you put them on some pseudo-time, right? Now at each bin, you have sort of a cell by gene matrix. So you take a bin, which is sort of roughly in the same time, if you will, and you can create this sort of gene by cell matrix, right? If you had a system which basically knew, if you knew the dynamical system, then you could have kind of found the Jacobian around each of these points. You can say, look, this is the state, I can find a Jacobian, right? But you don't have this, sadly, right? But you do have a relationship between Jacobian and covariance, right? This is the sort of famous Lyapunov equation or continuous time Lyapunov equation, right? Which is basically D is the noise, J is the Jacobian, and C is the covariance, right? Clearly knowing Jacobian and your noise, you can compute covariance, but the other way you can't do because covariance is symmetric and Jacobian is not, right? So you can't solve for Jacobian even if you know covariance. Good? Question? Yeah, this is assuming great. So this assumes that the system is kind of being slowly taken through from one type to the other, which again could be broken, right? So the signals are kind of coming in and dragging them through it. So you can think of, in this case, whatever the signal is, some kind of order parameter, if you want to think about some bifurcation, which is taking you through this. Right? So at each time, you're kind of adiabatically stable to this external perturbation. So then you can, but what happens near bifurcation is kind of, this simplifies a little bit, where so near a bifurcation, what happens is Jacobians have a zero eigenvalue, right? And there's an eigenvector along which it's kind of has that zero eigenvalue. And so if PD was that eigenvector, your Jacobian can be approximated basically with this one eigenvector where lambda d is this eigenvalue going to zero. So this equation kind of becomes simple around a bifurcation, right? And it turns out that also the eigenvector of C is exactly equal to the first eigenvector, which is not surprising to see now. I've made it a rank 1 matrix, so there's nothing else left, right? And the eigenvalue of this covariance matrix is blowing up, right? It's 1 over lambda. So eigenvalue of covariance goes to infinite, while the eigenvalue of Jacobian goes to 0, right? Because these things are multiplying to give you a finite number. That's another way of looking at it, right? So you can kind of use this, so like this is your 1D example, if you will, and we built sort of a higher dimensional example of this in just synthetic data, and you can see that you see this sort of eigenvalue jump up and go down precisely where you expect a bifurcation to go through. In this system, of course, we built it in by hand that the bifurcation was happening at some place. So you can kind of just look at it in this time series. Right? So now we're going to do the same thing for actual data and see what is possible. Right? Is the setup clear? So the two assumptions, but it's important that one of the key assumptions is this of steady state, right? That the system is kind of, the fluctuations in the cells that you're seeing are not, they're kind of kept together just because of sort of they are being taken slowly through something. So that's the big assumption. Yeah.
So there are essentially two steady states. So the light blue and the middle colors are not fixed.
So that's how I want you to think about it. So basically, it's saying that the system is always in a stable state, but it's getting dragged along something through a time-changing signal, right? So you can think of it, that signal is not changing fast enough compared to the internal dynamics, if you will, right? So for example, in this description, I put a S, where S is slow. Then you always reach steady state given some S. Right? That make sense? So yeah, here they are always in steady state. They're not two cell types.
The covariance is taken at one pseudo time.
One pseudo time, a bin around that, yeah.
So how sensitive is it to sort of wrong angle of lamination?
Yeah, so we tested all that in this data. You can basically, I mean, the key is you can't have too few cells. That's the biggest one. It's very sensitive to getting the pseudotime wrong, though. Right? So if you shuffle these cells around, for example, here this horizontal gray line is when you shuffle the timings around. Then you completely break the structure, as you would imagine. Right? So that's the most sensitive thing. So the bin you just have to not get the timing wrong. If you get the timing wrong, then all bets are off. Good? But the steady state assumption is I think, the biggest one. So in this, at least, data, you kind of see this quite easily. So you start here. So basically, pseudotime is from 0 to 122, however we came up with those numbers. And you can look at this sort of the first eigenvalue of the covariance matrix, right? And you basically see nothing. And you see sort of two features, if you will. You know, one of this, which kind of starts rising up, and the other is where you see this peak. And the simple models we built, again, I think there's more to be thought there, is you can see these kind of ramps when you have a pitchfork-like bifurcations, which are in some ways a bit fine-tuned. And you see these peaks when you have something like a saddle node, and you're switching from one, yeah. The gray is the shuffled here. Correct. So the reason with pitchfork is because you keep both the states. So one state turns into two. unless you separate the two cell types, you kind of mix that stuff together. So if you actually separate them out, you start seeing these peaks again. But for now, we can at least stick to this one, right? I'll have less to defend on that one, right? So the peak structure is definitely very natural, which kind of goes away when you shuffle stuff around. Yeah.
So in some sense, this is basically saying that the variability is the highest at the best.
That's all it's saying. Right? It's basically saying that when you make a sloppy mode, along the same direction, you're going to get variance. Right? That's what this equation means, basically, in the end. Right? Right? And now you can also kind of use this first eigenvector as I was speaking. And that also sort of fractionates very nicely these sort of three structures. So now what you're doing is you're taking your covariance matrix, picking a vector somewhere here, picking a vector here, and picking a vector here. So all 3 points, you can get a covariance vector, right? And you can make sort of a dot product of that around everywhere. And you see that sort of this picks out these 3 little blobs, right? So there's definitely structure which pulls out from this kind of analysis, right? We did a little bit more. This is, now you can kind of pull out, so as I was claiming that we think there's sort of a pitchfork here, so we can actually pull out these 2 cell types as they are being made at the first bifurcation, but we keep both of them, and then one of them apparently goes through this saddle node. So that's sort of the final answer we got to in terms of connecting these two. So if you, yeah, so that sort of is the structure, and this black line was the one which you saw for the whole thing. This is for the whole thing, yeah. Yeah. Oh, in this case, I don't know, right? I don't know how to separate that out from other, because there's also issues which I think, you know, Sean and Ashishman are thinking about is basically you can have many correlations because of lineages and things like that, which we couldn't really take them out. Actually, this data would have been good because they actually had some lineage information. I wasn't deep enough, but we weren't even thinking about it. So, you know, maybe there's a chance to look at it as well because you guys are thinking about it. We didn't think about it, so yeah. So here you can actually separate out these populations. It turned out people see them in experiments and they know these cell states, so you can kind of look for them. And again, you look for these along these slow directions. So along these slow direction, these populations separate out. Otherwise, they're all mixed up if you don't look at this particular eigendirection, right? So on that eigendirection, these 2 populations will split out, and then you can kind of do this analysis. So, I mean, roughly what this says to me is, you know, there are 2 big ifs in this, right? Pseudotime, how you build pseudotimes is a bit of a mystery. But it looks like at least it preserves something about ordering. It's not a real time. It's not telling you how long it takes, but it preserves the order. And that's important for looking for these things, or if they are there. And the second is this idea of steady state. So I think our hope was maybe to try to do this across all different cell types and, you know, really make a map of this sort of, you know, one Archishman showed in some ways of, you know, these saddles and fixed points or whatever. We haven't done it. But that could be an interesting way to look for things beyond just one of these branches, yeah.
In principle, these kinds of plots are made with the same kind of data. Why don't you see the bifurcations directly in this representation? Why do you have to go through this pseudotime and then covariance, and then somewhere over there's a bifurcation? You don't see it at all.
So here, because the way these are done, if I understand, is you basically define cell types, right? So you take, so in some ways, you're basically looking too far and you don't really see them because you're not defining in that fine-grained way, if you will. That's how I think about it, right? And that gets also very sensitive because you can keep splitting in many ways and it's not obvious how to do that. So the time ordering helps in that sense, right? So here, you're not looking at the time order when you're doing this, right? This coloring is not time ordering, right? You're just kind of just putting them together, right? Is that good? Yeah.
So if I remember they're actually already committed.
Yes.
So they actually have a...
Good. So I think that the main finding was also that if you look at, because they were doing lineage tracing, right? So if you go here and put dots in terms of this cell has this barcode, this cell has this barcode, some of those barcodes showed up more here versus on the other side, for example. Absolutely. Yeah.
I was wondering, I mean, how does that fit with this bifurcation? It seems that on that line, basically the cells are not deciding. They're already committed to one lineage.
Yeah, so whoever decided to come this way, that could be, in a sense, funneled for other reasons. I don't know, signaling or preconditioning of, epigenetic mark. I mean, at least the claim is that there are epigenetic differences, but I don't understand that biology enough. But it seemed like a continuous sort of scale in the sense that they're not still dedicated to any one of them. You see biases, right? So one lineage will show up more in these cell types compared to the other ones, rather than sort of exclusive. Does that make sense? Right? So it's not committing fully in that sense, right? So it's biased. And that's what we've seen in macaques also, early work we did now 10 years ago, where you can actually, again, do this barcoding, put it in macaques and look for what cell types they make. And you see some cells are sort of biased towards more granulocytes or more myeloids, but they're not committed in the sense that they will not make the other one. That happens later. I mean, here, not in this experiment. If you're doing good dish experiments, one could do that, probably. Yeah, and if you're kind of imaging them or something, right? Because these are all sort of taking a sample and then throwing that away, right? So yeah, that was one goal we had. But we haven't done that, but I think it could be interesting to do that if one was interested in mapping out the whole thing in some ways. You could also do sort of across organisms for same tissue. This is something one of the students is trying to do, is you can take different tissues and you could in principle ask, because in different organisms you don't have some of these lineages of cell types, right? Do they come from the same ones, which, do they come sort of near, they split off at the end somewhere, or they come from the source? Maybe one can ask those kind of questions and it might be interesting. Okay, so I guess in the last part, I want to kind of talk about, we got a little ambitious now. So now you have these lineages, right? So, and claim is how to build them, right? You can find special points in this. Now the question is, can we build sort of the whole tree structure of who goes to where? As Mukund was asking, you have this information already, right? Can you kind of use that to build it without sort of, going along one path at a time? And this was sort of work done by Spencer. So the idea was, now you have a lot of these single cells, right? We have some sense of that they get arranged in some way. The point is, can we learn sort of the dynamical system which is taking these cells through, right? That's the hope, right? Not just a bifurcation point or something, is there a dynamical system this system is giving you that you can learn from this data, right? This is a bit hacky, but what we did was, again, hope is that this is not happening in 100,000 or 20,000 gene expression space. Maybe it's a little bit shallower space. So we kind of, what we did was, as Sean was describing, sort of a variational autoencoder scheme, right? You can take this large sort of space of RNA, bring it to a low dimensional space, and ask whether you can write a dynamical system in this low dimensional space. Right? Yeah. Yeah, I'm going to say, right? So it turns out it's important to, and we were using sort of these neural ODEs, which were, came out now. Very old paper, I guess, 2018. Ricky Chen and David no. So again, if you remember basically, right, how gene expression happens is that you transcribe and you make these things which are called unspliced. And an unspliced RNA is then spliced and then either degraded or turned into a protein. And degradation is kind of saying it's gone out of the system, if you will, right? So again, as I was mumbling, right, so you have each cell, which it tells you the state space, but I think having this splice and unsplice helps a lot because it gives you a little local direction to use, right? So when we were trying these kind of schemes without including this splicing and unsplicing, we actually did not get good answers in the sense that it changed every time you try because you have a lot of room so as I was saying, you go in the local neighborhood and find a cell, right? If you find a few cells which are close enough, how do you figure out which one are you going to pick? But if you have a vector, it kind of breaks that symmetry and you can get it, right? So I think having this splice, unsplice information was hugely useful for doing this reconstruction. And you can actually build this into the dynamical system, because you can, rather than writing down one equation, you can say actually there's unspliced, there's spliced latent variables. Both of them are important, right? And they relate to each other in a very simple way that, so the unspliced thing, of course, never sees the spliced thing directly, unless you have a regulation variable, right? So the goal here, remember, we didn't only wanted to build trajectories, we wanted to ask, can we find out this self-scheme that the cells are using to guide themselves into different states, right? So we wanted to kind of learn this regulatory variable from this, not only sort of parse them into different types, but what is the structure of this regulation which is allowing these cells to become different, right? And in principle, the whole dynamics here still might be in reasonably high dimensions because it's, describing the whole trajectories, but the hope was that this is fairly low dimensional in some ways. So in most of our data sets, we found that the ZR could be like 3-4 dimensions, right? While this other Z's have to be 20-30 dimensions to explain things. Splicing vector is same as the. No, this is basically giving you some time ordering, right? At least the canonical model is you get unspliced, you get spliced. So if you see genes that are already spliced, that means they have happened at a later time than the genes that are not. So now if you see a new cell where you see more of the spliced compared to the unspliced, you know that must be closer to it than the other. That's the simple idea. That's the simple idea, right?
I don't know if this is too technical for
Yeah.
me, but how exactly does this dynamics, how do you impose it?
Yeah, so we impose it. Okay, I have one more slide, a little bit more, right? So how you impose it is very straightforward in machine learning language, in the sense you basically are finding, so each cell needs to be given a time now, right? Because we don't have a pseudo time assigned to them. So each cell needs to be given a time where it probably is in the system, and need to be given a vector z hat, which is this in the low dimensional embedding, right? And the goal of the dynamical system is to start at some place, z0, and reach that cell state at t hat time, right? Because this cell I've assigned a t hat time, right? So now my dynamical system better take me to this t hat, this state z, exactly the same amount of time that I've assigned this cell to be in, right? So you kind of sort of minimizing this function, right? Where t hat is assigned to a cell, and z hat is the state that the embedding is giving you, right? That's what your dynamical system should reach at. Make sense?
So you have to assign a t hat to every cell?
Yes.
So you have to assign the pseudo time before.
You're learning it.
Oh, it's being learned.
It's being learned at the same time, right? It's part of the autoencoding. So you're learning the parameters which also assign t hat. So you're kind of shuffling them around. But if you get the wrong order and your dynamical system can't kind of reach it, that means you're going to shuffle it back, if you will, right? That's the intuition. You're learning sort of t, and that you reach it at the same time. So that was I think, constrains the problem fairly well, I think. Yeah.
So you're making the assumption that it's giving you a time. It doesn't have to be.
Say it again.
In principle, there are many different processes going on.
Yeah.
But just if you increase of one or the other in spliced or unspliced, does it necessarily mean that?
No, it doesn't. No, it doesn't. It doesn't. So that's why there is no structure in these equations of saying one way or the other. They are just function of these variables, right? So I should have made that point, I guess. I mean, there are other models where people write parameterized model for these things and learn those parameters. Okay? There you, I think, make more assumptions of that nature in the sense. So here we kind of give up on that because also those models kind of don't capture these various states where you see, one thing happening first. So this is sort of the formulation of the problem. And the whole scheme of variational and autoencoders, as Sean described, you have all those elbows, you do your priors, you do your sort of usual machine learning tricks. So I think the only thing which is useful is I think this structure was immensely important for doing this, right? So you can kind of add some good amount of biology when you're trying to learn these things. So that was kind of useful. So I'll come back to this example of reprogramming. So this is how, as somebody was asking, so people did some single cell experiments where you start with these skin cells, these fibroblasts, and some of them reprogram, and you can see that they sort of can be stuck in these various stages for very long times. Right? So this is sort of a group which did reprogramming in a dish and did this sort of single cell sequencing and asked how does it look like. Right? So the question was, do we learn something more when we do this latent embedding, and can you see these dynamical systems in some ways? And what we see is kind of in this little space, this is sort of, in this case, there was just one-dimensional space, which explains most of it. So here, these are all the initial cells, which are here. And you can see that this initial variability matters because cells which are starting at this end actually turn out to reprogram more often than the cells that are starting at the bottom end. Right? So this, I guess, Ashishman, you were asking a question, right? Is this sort of built-in inherent early time dynamics, right? But I think you can already see that this sort of split happens a little bit later in time, and you can kind of get more. So here it's sort of like a unimodal distribution going into some bimodal distribution. That kind of problem is getting solved, and the ZR tells you precisely how it's doing it, right, in this problem. So of course, ZR can be mapped to the genes outside, right, from the decoder side. So we can actually tell which genes the ZR maps to in some ways, right? So now you can do perturbative experiments to ask if that changes this particular scheme one way or the other, right? Which way can you increase reprogramming or not? We haven't done those experiments yet. So yeah, as I said, the ZR in this case is just one-dimensional, right? So I think that, to me, was the interesting thing, that you actually have now one-dimensional dynamics along this time where you can, if you're only interested in 2 states, perhaps you don't need more than that, right? And we were trying to do this with bigger systems like 20 states, and then ZR needs to be big. You're embedding more states. But you can see that you can actually bring it down quite considerably, and then not so much, right? You kind of get stuck at some number as you start bringing this ZR down some ways, right? So that was the goal of trying to find sort of the smallest system which describes this kind of dynamics. And I should say one has to be careful. You can say, well, do you now start predicting a lot of important genes? I think one should be careful because you embed so much. I think you're going to lose a lot of information about individual genes, particularly the genes which may be lowly expressed or noisy. So I think that is still hard, but it gives you something about what kind of dynamical systems might be interesting to do this problem. I still have a few minutes. We applied this we applied these ideas to with some local people on lung development. I think there's more biology in it than I can get to. But what you start seeing is, I'm going to just not say too much about it. You start seeing is you can actually see multiple paths of reaching the same cell types at different time points. So apparently the same cell is made through different paths as time goes on in development. So you can kind of tease out these temporal dynamics a little bit easier, and then you can ask some questions about it. I think I can probably stop here. I've thanked most of the people. Maybe as an outlook, I think what we want to do is really go back to this reprogramming problem, and we are working with a local group where we're trying to deeply do this lineage tracing with gene expression, so we can really tease out this idea that how much of it is coming through signaling versus intrinsic, and what is there sort of development-like pathways as these things are going back, or is it sort of a bit of a search problem? And the second part, which I'm excited about, is really trying to understand the properties of these dynamical systems that we learn. So for example, when you look at these dynamical systems and you ask whether they have steady states, they don't, right? If you just set fz is equal to 0, you really don't find many steady states. And in some ways it's not surprising, probably because these tissues are still in flux, right? These are developmental tissues and they're still getting made. So it's not that the cell is stopping somewhere in some ways. I'm not looking at the end point differentiation yet. And you can even see in individual genes that they're still ramping up. They haven't become steady state in that sense. So you can see that structure. So you don't immediately find this steady state structure? Do you see sort of funnels and things like that where cells flow from one type to the other? We haven't looked at that, but that's what I would be interested in looking at, the sort of the topology of these dynamical systems. Can we think of them as saddles and attractors, and or you need a little bit different structure to think about that? I think I can end there. Thanks, sort of people. It's old photo of a group, but yeah, so the people were in this. So thank you.
Is it possible to incorporate domain priors by way of these Yamanaka patterns or these classical patterns and talk about controlled ODEs instead of regular ODEs, because they kind of constrain the space of regulatory factors that can impact this evolution.
So you can build a lot of structure, which I didn't show, which we tried a lot, is basically in these sort of last equation, for example, you can build a lot of regulatory structures. You can even add things like chromatin information in this. You can bias by saying, which chromatin is open or not, and that can constrain what is available in the gene space for it to work. And you can even encode cell types if you want in this information. And timestamps when you have them. You can enforce the things that are in same time. If it's a very temporal process, you can also regularize that in some ways. So you can build those correlations quite easily in this framework. Yeah. And in fact, we used this data to kind of predict what attack seek should look like, and then compared it to it, and it looks actually pretty reasonable. This paper, yeah.
Middle part of your talk, I'm trying to
Good.
So you have these multiple types of cells implicitly in there.
Yeah.
Somebody's made the assignment of different cell types, but you don't use that.
Right.
Somebody's made an assignment of pseudotime.
Right.
My question is, so pseudotime is somehow local in this space.
Correct.
Correct. But it's not two-dimensional, right?
No. It's in some, yeah, exactly. So I'm just using time information of that, yeah.
But it's pseudotime. So my question is any smoothly varying function, local function in the space going to give you these peaks? Does it have to be pseudotime? Does it have to match with real time?
No, I think the ordering has to be correct, as I said.
I'm saying any smooth cut across the space.
It's a good question.
You did an experiment where you shuffled totally the
Yeah.
Any smooth curve will occasionally be aligned with some sort of pseudotime. So is that what you're seeing, some purely geometric feature, or does it allow you to say these are true pseudotime deductions and these are not?
I mean, I think what I can say for sure is that these kind of peaks would not emerge in a random slice, I think. Right. Smooth.
So smooth in this space. I'm not saying random.
Okay.
Smoothly through this high-dimensional space where there is a locality condition.
Right. Then it would work, I think, yeah. I agree with that. Yeah. I think if you have some ordering correct and then you slice it, I think it'll work. I think that intuition seems to be correct. I think, yeah, I agree. I know, I totally agree. Totally agree.
Is it just to do with how you model the signal?
I don't know. So here we basically, there's no model, right? Here there's no model. It's basically just saying, look at the covariance matrix. Does it have structure, right? The other one is probably trying too much, right? It's probably trying to sort of get the whole trajectory of things. And in that, I don't know. I agree with that. I mean, okay, let me say it this way. It's not obvious to me here these bifurcations, so they're steady states in a very different way, right? So if you really wrote down a dynamical system here, you might not find a steady state because it's still flowing, right? All I'm saying is that this direction is kind of slower than the other direction, right? So here I'm just relying on the separation of time scale, right? So I think that's the best I can think of right now, that, it doesn't fully break it in the other model. Yeah. Yeah.
Maybe they're not conflicting because your bifurcation are a zero eigenvalue bifurcation, right?
Okay.
And if I have a zero eigenvalue, it does not necessarily need to describe a bifurcation. It can also describe a slow state.
It can describe a slow state, yeah, for sure, for sure, for sure. It's just yeah, that's another way of thinking about it, yeah. But my money is more on the other one in some ways. It's kind of sort of, it may be sort of this adiabatic thing which is kind of getting dragged along. So you won't, it looks like you don't see it, but. Questions? Okay. Cool.
If there are no more questions, I would say we can break for tea,
Yeah.
and then we come back in sort of like 37 minutes for the around then for the discussion.
Yeah, okay, thank you.